Hello guys, a very warm welcome to one and all present here. Myself Harshali Awai, roll number 6203 from SYBSC Ramnaran Junior College. Today I am going to give you a short overview of one of the important techniques in microbial taxonomy called the fame analysis technique. This technique comes under the chemical analysis. So let us see what this fame analysis is and how it is actually done. These are the contents of my presentation, so we will discuss it one by one. The first question which comes into our mind by listening this name is what is FAME? So FAME stands for fatty acid methyl esters. These fatty acid methyl esters are a type of fatty acid ester that are derived by the transesterification of fats. Now what is transesterification of fats? When a fatty acid reacts with alcohol in presence of base catalyst, it gives glycerol and the respective fatty acid ester. In this case, the alcohol used is methanol, as we want to get methyl esters. So it requires an alkali catalyst. It can be sodium hydroxide, sodium methoxide, or potassium hydroxide. Let me introduce it. As we all know, lipids are the major constituents in a cell, as it are present in lipid bilayer and also in lipopolysaccharides. This makes them one of the most useful biomarkers for the identification. So there are two common approaches of analysis of microbial lipids. One is polar phospholipid fatty acid analysis and the second is total fatty acid methyl ester, which is called the FAME. We are going to discuss the second one in detail as it also includes PLFAs. So let us see the objective of this analysis. As we all know, every microorganism contains certain lipids and fatty acids. So these fatty acids are esterified and then they become volatile enough for the analysis with gas chromatography, which is used to create the FAME profile. As FAMEs are more volatile than the fatty acids, we can easily detect it by the gas chromatography technique. So every microorganism has its specific FAME profile. It can be used in microbial fingerprinting. These profiles can be used as a tool for microbial source tracking to identify the bacterial strains and also for characterizing new species of bacteria. Like for example, if we make a FAME profile of a newly cultured bacteria, by analyzing the results, we can compare it with the known bacteria and we can understand if this is the earlier known bacteria or a new one. So this FAME analysis is done by gas chromatography. The main reason being the FAMEs are more volatile and thermally stable than the fatty acids. And as we all know, gas chromatography has become a very important technique in fats and oil analysis because it gives accurate results obtained for complex as well as simple sample matrices. So let us see the procedure of how this technique is actually performed. So first, from the cultured bacteria, the respective fatty acids are extracted and followed by the derivatization with dilute HCl and methanol to give the respective methyl esters, FAMEs. What is derivatization? It means it is basically a chemical term which means producing a new compound. So as from fatty acids, we are producing methyl esters. Then the FAMEs after extraction, they are extracted from the aqueous phase by use of an organic solvent. Then these FAMEs are injected on the gas chromatography system for separation and are subsequently detected. This is done by a software called the Sherlock software. Now, what is this Sherlock software? Sherlock software was launched in 1991 and it has a rapid, accurate, and inexpensive solution for the identification of over 1,500 microbial species, fatty acids, and PLFAs by GC-FAME analysis. So this is basically a library which has stored entries for identification. This software automates all the analytical operation and uses a sophisticated pattern recognition algorithm to match the unknown fame profile to stored library entries for identification. This makes our job more easier. And this software is capable of identifying a wide range of microorganisms, as this library was carefully developed by collecting well characterized strains of reference culture from microbiologists in many areas like clinical, environmental, industrial, drinking, wastewater, and food sample. There are two types of Sherlock softwares. First is called the iFame and second is QFame. These are nothing but iFame is most useful for edible oils, marine soils, and biofuel feedstocks. It has the information of all these fatty acids, and the QFame is used for the aerobic bacteria. iFame takes less than 15 minutes for identification, and QFame takes less than 25 minutes. This was just more information on Sherlock software. This is a pictorial representation of how the technique actually is. Once the culture is taken, then the fatty acids are extracted, methylation is done in order to get the respective fames. Then these fames are also extracted and injected into the gas chromatography system. Then GC analysis is done, we get respective peaks and the results are then compared and identified by the pattern recognition software like Sherlock software. These are the results. In results, we can basically see the peaks are obtained for different fatty acids. The x-axis shows the time and the y-axis shows the amount of fatty acid. By this, we can easily recognize the fatty acids, and these results can then be compared and detected for the identification of bacteria. The rest of the job is done by the Sherlock software. So this advantages of this analysis. This technology is very accurate as we have this automated system. Also, it is inexpensive technique comparatively. Fatty acid analysis is an effective tool for strain tracking and also advantage over other genetic identification system. Beside that, all microorganisms catalyzed with the same procedure. And as fames are more volatile than their respective fatty acids, it can easily be detected by the GCA analysis. These are some advantages of the fame analysis technique. Thank you. I hope you must have understood what is this technique, like the overview and how it can be done.